Good evening, everyone. Hope all of you have enjoyed and learned a lot from our previous sessions. So, finally, we have reached to the ultimate day of RACB 22, which will be definitely as interesting and informative as the previous ones. It is really a privilege to get such an audience whose constant interactions made the webinars livelier. Hoping to get such responses today as well. Today's webinar will be conducted by Dr. Ajoy Basak of University of Ottawa, Canada, whose lecture on protein aggregation in Alzheimer's dementia, what do we learn from research, will definitely mesmerize all of us. So, without any further delay, I would like to call upon Dr. Reena Ghosh, Associate Professor, Department of Chemistry, St. Xavier's College Autonomous, Kolkata, to introduce our speaker for the day.
Thank you, Shaumitri. Good morning, Dr. Basak, and a very good evening to my dear colleagues and my dearest students. On behalf of the college, sir, I would warmly welcome you to St. Xavier's and the Department of Chemistry, this time on a virtual platform, and look forward to having you on board in the near future. I also extend a warm thank you to our department chairperson, Dr. Onkur Ray, for choosing me to chair this session. It's an honor and a pleasure for me to do so. Now, regarding the speaker, I would like to acquaint my colleagues and everyone here, my students, about the achievements of Dr. Ajoy Basak, who is the adjunct professor, Faculty of Medicine, University of Ottawa, Canada, and a specialist in protein chemistry and enzymology. He did his BSc in chemistry from Presidency College, and he obtained his MSc degree from Calcutta University with organic chemistry as specialization. Of course, now I again tell you, my dear students, don't think of specializations because Dr. Basak himself has crossed barriers and has tried to merge, all our conventional compartments of chemistry. In 1975, he received his PhD degree in medicinal and natural product chemistry from Bose Institute, Kolkata. He is an extremely experienced research professional with more than 20 years of experience in teaching and supervisory experience in health sciences and bio and medical sciences as well. He has a strong background in analytical chemistry, enzyme chemistry, peptide synthesis, drug design, and delivery. He is an eminent professor whose teachings involve proteins, peptides, protease regulators, and natural proteins, natural products of interest. This eminent scientist has a huge contribution towards science, especially in the field of biological and chemical sciences, which we really cannot express. He's also a member of certain prestigious societies like the American Chemical Society, Canadian Institute of Chemistry, Indian Chemical Society, Institute of Chemists. I think the list is endless. So let us sit back and listen in all eagerness to you. And so students, we gear up and you also gear up to the presentation of Dr. Basak. Over to you, sir.
Thank you. Thank you very much and for your nice interaction. And it is really a great pleasure to give this lecture. I hope that I could be there physically. I had been to St. Xavier's College. At that time, it was a college. And I had the privilege of giving this lecture physically there. And it is really an interesting way of communication now. But I hope that I will keep you engaged in this talk. And let me share the screen. Can you see the screen? I hope you can see the screen, right?
Yes, sir. Yes.
Okay.
Screen sharing is on, Dr. Basak. Yes.
Okay, thank you. So I will go to the um, all right. So today's lecture will be on protein aggregation in Alzheimer's disease. This is a topic that we have been working, and we have published quite a bit of papers on that. And during the past 30, 40 years, there are a lot of research has achieved in this area, and we have known a lot of things, but still there is no cure for this particular neurological disease. Now, before I jump into Alzheimer's disease, I would like to give a little bit of background of the neurological disease. As you know, that this is a brain disease, and that disease could be structural type, or it could be it could be biochemical type, or it can be electrical type. Electrical type means there is some abnormalities in the electrical communications, and the consequences are endless. It can be paralysis, it can cause muscle weakness, poor coordination, memory loss, seizure, confusion, and so on. And the question is, what are the interventions? As I said, there is no cure for this, so we can only manage the disease with some kind of medications. And some medications are available. So at the very end of the lecture, I will provide you some of the drugs that are being currently used in order to keep the disease under control. So here is a brain. This is only a 2-pound weight material of tissues consisting of the neurons or nerve cells. And basically, brain can be subdivided into 5 separate sections. And they have their names. We can call frontal lobes, temporal lobes, occipital lobes, but parietal lobes, cerebellum. Now, all this is connected ultimately to the stem, the brain stem, which is here, and that controls our breathing and blood pressure, heart, and also swallowing. So you can see that each section have their own particular roles, and these are all listed in these little boxes. And the very back, which is the cerebellum here, which is a very small area, but that controls balance, movement, and coordination. And this can be the cause, if there is some dysfunction in that area, that can be the cause of what is called Parkinson's disease or motor dysfunction. But I'm not going to talk about Parkinson's disease. I'll be talking mostly about the Alzheimer's disease, which is a dysfunction of the frontal lobes. So what is Alzheimer's dementia? Now, it is on the top of the list. As you can see, that there is at least 23 different neurological diseases that have been described and have been heavily researched. And there is a one common feature in all of these dementia, is that there is one or multiple proteins that have been implicated in a way that these proteins somehow gets self-aggregated, self-assembled, and that leads to the formation of plaques or protein precipitates in the brain. So let's go through these diseases. You know Alzheimer's disease, and in this disease there are 2 proteins that I'm going to talk very extensively. One is called amyloid protein, and another is called tau protein. And these 2 proteins are the cause for Alzheimer's disease. I'm saying cause means that these proteins dysfunction and they get precipitated in the brain, and that blocks the neuronal messages, and that creates all kinds of dementia. Then the next disease of interest is Parkinson's disease, which is a motor function. That means you lose your balance. There is a tremor, uncontrollable movement of hands, legs, head, and many parts of the body that you cannot control. And what is the cause? The cause is the Parkin protein. And Parkin protein is also another large protein which gets precipitated in the brain. And the list goes on. There is a Huntington's disease, there is a mad cow disease, which is called CJD disease, and this is the human version of the cow disease. And this is also linked to another protein called prion protein. Now, the word prion means toxic, and a lot of proteins become toxic as a result of aggregation and precipitation. And then there is ALS, which is also called Lou Gehrig's disease. And these are all very, I would say, uncurable diseases. You cannot cure them. So now all these diseases have been very thoroughly described in the literature. The human brain tissues, the mortal brain tissues of these patients have been examined histochemically, that means using microscope. And these are some of the pictures of the microscope that I want to share with you. One is called fibril or rod-like structures. This is a feature that you can find in Alzheimer's disease brain. That means in the brain, there is some what is called plaques. That means precipitate. That means condensed form of proteins that are precipitated gradually over many years. And that is one of the major causes of the dementia that we see in Alzheimer's disease. Now, there are many kinds of protein precipitates have been found in other neurological diseases. For example, there is a kind of what is called spongy form. That means this is a precipitate of protein where you can see some holes, like your sponges. Inside the sponge, you have many holes. So in the brain, there is a protein that is precipitated in the form of sponges. And then some proteins are precipitated as a very compact form, very solid and compact form, condensed form. So there are many ways, many shapes and morphological features of these protein precipitates. Now, now here is another schematic diagram. This is a artistic diagram. This is not the real picture, but I can give you some idea what happens in the brain. So you can see that the blue color indicates that it is a healthy brain, healthy neurons. Now, when it becomes white, that becomes you are losing 20% of the capacity. And you can see as the disease progresses over a period of 18 months, the white portion of the brain becomes more and more, it increases. So suggesting that there is a loss of neuronal activities in the brain. And this disease has been discovered more than 100 years ago by Alois Alzheimer. And he was studying one patient, and patient's name is August. And this is in 1906. And just about 14 years ago, we celebrated the 100 years of Alzheimer's disease. Now, this is the real picture of a mortal tissue brain. So this is the plaques that you see. Plaques means these are protein precipitates in the brain, and these have been stained with silver iron. Now, silver iron is a metal ion that can stain protein precipitates, protein aggregates. And so this particular reagent has been extensively used along with many other dyes, which can detect the formation of protein precipitates in the brain. So now if you blow up one of these condensed material, it will look like that. And in the middle, this is a huge condensed protein precipitate. And along this precipitate, this is called plaque. And along this, surrounding this precipitate, you have what is called fibrils. And if you blow up this part, it will look like rod-shaped structures, long rod-shaped structures. So these are all protein precipitates. But around this is actually tau protein around it. And in the middle, you have your amyloid peptide precipitates. But the main feature is that there are two proteins in Alzheimer's disease. One is called amyloid beta and another is called tau. And these proteins somehow they get aggregated. And that means they self-assembled, they interact with each other, and they form dimer, trimer, tetramer, and who knows how many mers are there. So it is a huge polymeric peptide or protein that are formed in the brain over many years. So this is extremely slow process, but at the end, you have an extensive amount of protein precipitates that will be formed in the brain, and that will cut off all communications from one neuron to the other. So remember these names of these two proteins. One is called amyloid beta, and another is called tau protein. So I will go through the individual proteins separately. So in the beginning, I will talk about amyloid. Now, I sometimes call them as peptides, sometimes I call them as proteins. As you know, the proteins are nothing but peptides, but very large peptides. So from 2 amino acids until maybe 50 amino acids, we call them as polypeptides or just peptides. And if it is beyond 50 amino acids, we call them as small or short peptides. And if it is beyond 100 to 120 amino acids, we call them as full length proteins. So we sometimes switch the peptides and protein words very often. So it is just the size, that's all. Now, A beta or amyloid beta, it is actually a peptide because it is only 42 amino acid long. So 42 amino acids should be called peptide, it should not be called protein. But it is coming from a very large protein, which is called APP, amyloid precursor protein. And this amyloid precursor protein is naturally present in our brain. And there are 3 enzymes that I will talk about. One is called beta secretase, another is called alpha secretase, and another is called gamma secretase. I will discuss about these roles of these enzymes in the formation of A beta or amyloid beta peptide, which is coming from APP following cleavages. Now remember, these are all proteolytic enzymes, meaning they cut large proteins and form small peptides. So large proteins, for example, here, APP, this is functionally inactive. That means it has no function. However, when it is cut or cleaved by these enzymes, it can produce peptides. And those peptides will be called bioactive peptides because they are now functionally active. Now, here is a schematic diagram of amyloid precursor protein. Remember, this is a protein, which is a very large, it is 770 amino acid long protein, and it has a membrane span. So this area is bound to the membrane, cell membrane. So there is a huge portion, this portion is intracellularly located. That means it is inside the cell, and this part is outside the cell. Now there are 3 enzymes I mentioned, beta, alpha, and gamma. And you see the sequence. I did not say alpha first, beta second, gamma last. So I say beta, then alpha, then gamma. And you will see the implications of these. The implication is that when beta cleaves here and gamma cleaves there. Now I put 2 gammas. One is called gamma secretase, creating a 40 amino acid long peptide. Another is gamma secretase, creating a formation of 42 amino acid peptide. So there is a controversy. I should not say controversy. There is a lot of research that have predicted that there are 2 types of cleavages here. So either you can produce 42 amino acid or you can produce 40 amino acids. From here to there, you have 42. And from here to there, you have 40 amino acid. And this cleavage occurs within the membrane. And this is intracellularly happening. So when you have a beta and gamma enzymes cutting APP at 2 different places, you produce what is called A beta peptide. So A beta peptide, which could be either 40 amino acid long or 42 amino acid long. And normally we get a mixture of both. And so you are getting a mixture of 40 and 42 amino acid, which we now term as A beta peptide. or amyloid peptide. And you can see that in this pictorial diagram, this is a beta cleavage, which is intracellularly, and this is a gamma cleavage, which is happening inside the membrane. And that creates your A beta peptide. Now, what is the significance of A beta peptide? Because this is the real cause of Alzheimer's disease. Because this A beta peptide, which will show here schematic diagram. Now remember, this is a 770 amino acid long protein. This is a TM, means transmembrane domain. So this is anchored to the membrane. And there is some cleavage, which is done by gamma secretase happening within the membrane. And another cleavage is here, which is the beta secretase cleavage. So when you have beta secretase and gamma secretase, then you can produce the A beta 40 or 42 peptide. So the formation of A beta 40 or 42 peptide is the first initial cause of the disease, but the real cause is we don't know what happens after that and what causes A beta 40 or 42 peptide to form self-aggregates, a large precipitate of polymeric A beta 40, 42 peptide. So there are many, I would say many points here to take a note. First of all, the beta gamma secretase activity are very high in Alzheimer's patients. That's why you have a huge amount of cleavage here and huge amount of cleavage here, and that leads to the formation of A beta 40 or 42 peptide. And after that, this peptide can remain in the soluble state, but for certain reason or certain factors influencing that lead to the formation or aggregation of A beta 40 or 42 peptide. Now, in normal people who doesn't have Alzheimer's disease, they have a separate cleavage, which is called alpha secretase cleavage. So the alpha secretase and gamma secretase cleavage, and that produces a peptide, which is called P3 peptide, and it is a smaller peptide, and it is not toxic, meaning that these peptides don't aggregate. So what is the difference between Alzheimer's patients and non-Alzheimer's patients? The difference is that in Alzheimer's patient, the beta gamma cleavages is very high. In the case of normal people, it is the alpha cleavage, which is very high. And if you have alpha cleavage, which is happening in between the A beta peptide, you cannot produce 40 or 42 mer peptide. So that is the one cause. That means the beta and gamma enzymes are very high in the case of Alzheimer's patient. Now, many mutations have been found in APP. And one of the worst mutation is called Swedish mutation. Now here you can see that this is the APP protein, and this is the membrane area. And there are three cleavages. One is here, which is beta, and here is the gamma, which is inside the membrane, and this is your alpha, which is a good cleavage. Good cleavage meaning that this will not lead to Alzheimer's disease because it cannot produce A beta 42 mer peptide. Now, in the case of some of the Swedish people, where Alzheimer's disease has been found very predominant in many years ago, and they were trying to understand why certain section of the people are so much prone to this particular disease. The reason is that mutation. There are a lot of mutation after that have been discovered, but Swedish mutation still remains the worst. Here you have a K means lysine, M means methionine. So this mutation makes this cleavage very facile. So that means you have a huge amount of A beta peptide produced and in a rapid fashion. And that is the reason for Swedish mutation. The people who are having this particular mutation, which is now called Swedish mutation, they are cleaved 9 times faster. So therefore, you are producing the protein aggregates 9 times faster rate and also large amount. And many other mutations have been discovered later on, but they are not as bad as the Swedish mutation. So the 2 points to take note here. One is the formation of A beta peptide, which is done by the cleavage of by beta secretase and gamma secretase. And the second thing is the aggregation of A beta peptide. And I will discuss a lot about this aggregation event and what are the causes of this aggregation as we know right now. So here is the list of all the potential causes. Now I say potential because some of them are still being proposed, and some of these factors are now being still researched. And there are certain connections is very, very high. For example, if the protein has a high amount of beta sheet structure, then that protein is prone to undergo self-aggregation faster. So it is the protein structure with a beta sheet feature, they will aggregate a lot faster. Second thing is that this is a metal ion theory, and this is one theory that there's a scientist, his name is Cox, and he first started this idea that maybe high level of certain metal ions in the brain tissues are linked to protein aggregation, leading to dementia. So there is a big connection between a high level of protein ions in the brain tissues with the formation of protein aggregates. And there are many other, which I'll not go through because of lack of time, but there is another area that I will touch, which is called neurotoxins. So there are certain chemicals in the environment, which we call them as neurotoxins, because they are toxic to neurons. That means they damage neurons, they destroy neurons. So I'll talk about few of those neurotoxins. So I will touch mostly about the metal ions, what is their roles. And so far there are a huge amount of studies that support this theory that high level of these particular ions are responsible for formation of metal aggregates. And these have been very well demonstrated by several studies, not only in Alzheimer's field, but also in other fields like Huntington's disease and Parkinson's disease as well. Now let's talk about the A-beta-42 peptide, because this is the major peptide that is formed in the brain because of the action of beta and gamma secretases. And here is the entire sequence of A-beta-42. I hope you are all know about the single letter code of each amino acid. And for example, D is aspartic acid, A is alanine, E is glutamic acid, F is phenylalanine, and so on. So you can go back and look at the sequence. And the main feature of A-beta peptide, A-beta-42 peptide, this peptide can be synthesized and it is available commercially. And we have made this peptide in our laboratory. And we also made many mutated A-beta-42 peptides. But one interesting feature of the structure of A-beta peptide is that there is a huge amount of sheet structure component in the middle. And this middle sequence is shown here in blue. And if you look at this sequence, you will see that there's a lot of hydrophobic amino acids. For example, leucine, valine, phenylalanine, phenylalanine. So these are highly hydrophobic amino acids. And these hydrophobic amino acids are responsible for sheet structure, and that lead to self-interaction and formation of aggregates. Now in this structure, you will see some blue circles here. These are basically water. So when you do the crystal structure of A-beta-42, and then it will look like that with some water molecules attached to it. Now, these are the several protein secondary structures that I have drawn here. You can have helix structure. Helix is very common in proteins and they have many roles in functions. But in this particular case, we will be talking mostly about the sheet structure, which is basically a flat structure, like a sheet of paper. And they can lie on top of each other through hydrogen bonding. And therefore, they can lead to the formation of large amounts of highly polymeric compounds. And then you have turn structure, loop structure, coil structure, and so on. So I will not go through all of them. So our focus is here because this is one of the causes of protein aggregation. And these secondary structures of proteins can be studied using two particular procedures or spectra. One is called circular dichroism. This is very useful. And this particular machine is useful for analyzing protein structure. And this will give you the information about what is the percentage of various structures of the protein in aqueous solution. Now, we have done this mass spectrum in our laboratory, and this is A-beta-42 peptide that we made in our laboratory. And its molecular weight is 4514. You can see that this is the molecular weight. But after 7 days, we see that it is forming dimer, which is this. This is a trimer, this is a tetramer, and this is a pentamer. You can see that it is going up to decamer. So up to decamer, we can detect in aqueous solution by mass spectroscopy. So mass spectroscopy is a very powerful tool for detecting the various polymeric forms of proteins. So this tells us that A-beta-42 is really very susceptible to self-aggregation. And this has been done in a pH of 7.4 in a buffer medium. Now, metal ion theory. Coming back to the metal ion theory, there are a couple of metal ions that I will focus mostly because these have been heavily linked: copper ion, zinc ion, and mercury ion. These are the three ions that have been linked to aggregation of protein. And these are some of the amino acid residues in A-beta-42, which have been implicated in interaction with these metal ions and promoting self-aggregation. And you can see that this area, you have histidine, which is basic amino acid, K, which is lysine, which is also a basic amino acid. And there is a participation of Y, which is tyrosine, and there is some basic residues here. So these couple of segments or areas that have been part of this self-aggregation, which is induced by these metal ions. And this is a very clear-cut support of this metal ion theory. You can see that in normal brain tissue, healthy people who are not suffering from Alzheimer's disease, and their brain tissues after their death had been analyzed. And we can see that in their brain tissue, the amount of copper ion per milligram of the tissue is 0.004 milligrams, which is a very tiny amount. But in the Alzheimer's patients, the corresponding value ranges from 0.46 to 1.76 milligrams. So clearly there's a huge accumulation of copper ion in the brain tissues of AD patients or Alzheimer's patients. Same thing is true for zinc ion. You can see the normal people, the amount is 0.0722 milligram per milligram brain tissue. Whereas in the case of Alzheimer's patient, the value is 0.12 to 0.93. And ferric ion has also increased. So you can see that there must be some connection because of these results. And in fact, these A-beta-42 peptide have been studied further in vitro, and they have found that this peptide can form complex with copper ion. So this is your copper ion, and there are 2 histidines and 1 tyrosine, actually 3 histidines. These 3 histidines and these tyrosine. This is in position 10, this is in position 6, this is in position 14 and 13. And they are involved through hydrogen bonding to form a copper complex. And people have also identified that this copper ion can form a dimer. And here is the dimeric form. So it shows that maybe the copper ion mediates in the formation of aggregation. And here is the area where the interaction with copper ion has been found to be maximum. And I have shown you that the two histidines here, one histidine here, and one tyrosine here, these are the 4 amino acid residues which are involved in the formation of this complex. So it is almost, I would say almost, because there are some people who don't believe this metal ion theory, while others are very supportive of this theory. And I believe that there is definitely a strong connection of these metal ions with protein aggregation. Now, here is a little video I want to show you, if I can. Here, you can see these black spots. These black spots are actually proteins, and how these proteins are coming close to each other and they are forming an aggregates. And you can see that, for example, here, this black spot is becoming larger and larger because they are interacting with one another. You can see here is a huge amount of aggregates are forming. Now, this is not a real thing because this has been done in vitro in a cell line. But this is what is happening in the brain over course of years. So up to this, I discussed about the A beta peptide. So the take home message is this, that in the case of Alzheimer's patients, the beta gamma activity is very high, and that can form a large amount of A beta 42 peptide, which is initially formed in soluble form, but then because of certain factors, it aggregates. And there are 2 factors which are very in the top of the list. One is the metal ions, and the other one is neurotoxins, which I will come back very soon. Now I'll move on to the second protein, which is called tau protein. Now I put the letter H in front because this is a human tau protein. And let's talk about tau protein, why it is so critical. What is the function of tau protein? Now, you all know that the brain tissues are made up of neuronal cells or nerve cells, and they look like this. In real, they look like this, but this is a schematic diagram. And this area, which is a long stem, and this is the axon area. And when this area is very strong and straight, then the neuron is healthy. And in order to make this stem very strong and healthy, tau protein plays a big role. And here are some microtubules. This has been expanded in this area. So this area look like this, and these are microtubules that are lining across this axonal length. And these microtubules will remain very strong, rigid, and straight as long as it is bound with tau protein. This is your tau protein. This is your tau protein. So in normal people, these tau proteins are strongly attached to these microtubules and making the axon very strong, rigid, and functional. So the neurons become healthy. Now, when these tau proteins are phosphorylated, here is the P, then it loses its binding properties. And because of the loss of binding properties, these axonal tube, which is formed by microtubules, they collapse. And as a result, the neuron dies. So this is the main, another reason for Alzheimer's disease. That means in the Alzheimer's patients, these tau proteins, they become phosphorylated and actually hyperphosphorylated. That means a lot of phosphorylation groups are there. And that leads to the collapse of the neuronal axon. And this is not the only thing that happens. These tau proteins also aggregates as a result of phosphorylation, and they form what is called PHF, paired helical fibrils. So these are the rod-like structures that I showed you in the Alzheimer's patient's brain. And these constitute the polymeric tau protein. So the main causes are listed here. You have huge amount of phosphorylation, which we call hyperphosphorylation, because normal people also have phosphorylated tau, but this is a manageable level of phosphorylation. In the case of Alzheimer's people, you have super phosphorylation. There is too much phosphorylation. And phosphorylation can only happen on serine, threonine, and tyrosine residue, mostly on serine and threonine. So somehow these residues are super phosphorylated. And what is the reason? The reason is the enzyme. There is the enzyme called kinases. The kinases are the phosphorylating enzymes. That means they put phosphate on serine and threonine. So somehow the kinase activity went up in the case of Alzheimer's disease. And the phosphatases, which remove phosphate or dephosphorylating enzymes, their level is low. So that means they cannot remove the phosphate. So it is the balance between kinase and phosphatase. And there is another factor that have been introduced recently that is called nitrating agents. There are 5 tyrosine residues in the entire tau protein, and these tyrosine can be nitrated. And this nitration is caused by some agents, including peroxynitrite. Where it is coming from? It is coming from environment. So therefore, if your brain or that person is exposed to an environment where there are a lot of nitrating agents like peroxynitrite, then the tau protein can be nitrated extensively. So nitration and superphosphorylation, these are the 2 causes of aggregation of tau protein and loss of binding of tau protein with microtubules. Now, this is the entire structure of human tau protein. I don't want you to go through each and every part of it, but I would say that here is a threonine T, and here is a serine, and here is another serine. Those are the 3 amino acids which are highly phosphorylated in the case of Alzheimer's patients. And they have been linked to one of the causes of loss of binding property of tau with microtubules. And I have shown you some boxes here. There are 4 boxes, one here, this is 2nd, this is 3rd, and this is 4th. So these are the area via which tau protein binds with microtubules. Now, another thing that has been recently observed is that the tau protein is also cleaved. And this cleavage is done by an enzyme called caspase-3. So caspase-3 is an aspartyl protease. So that means it cleaves after aspartic acid. And this is the area, D is aspartic acid. So this is the area where caspase-3 cleaves the 441 amino acid long human tau protein and make it 421 in that case. And this shorter version of tau protein has been found to be large amounts in AD brain. So therefore, this cleavage by caspase-3 is also critical. And we have demonstrated using mass spectrum and using some peptide models that yes, it is cut at this particular site by caspase-3, and it can produce 2 fragments. So this is not the whole protein, but this is a small peptide, which is derived from the cleavage site of tau protein. And this has been also detected in the brain that the shorter version or the truncated tau protein is in high amount in AD brain. So this is the story about the tau protein. That means the tau protein, there are 3 things happening. One is the phosphorylation, superphosphorylation. Second thing is nitration. And 3rd thing is cleavage by caspase-3. So all 3 events have been linked to the development of Alzheimer's disease. Now there is another area where a lot of people have put a lot of attention, that is the neurotoxins in the environment. So one of the neurotoxin, it's a very small molecule, it's called BMAA, which is beta N-methyl amino alanine. So it is an amino acid, but it is a special amino acid, unnatural amino acid. There is a methyl group on alanine. So alanine is a natural amino acid, but this is a methylated alanine. And this is called BMAA. And this is the molecule. This is an extremely small molecule. And it is found in cyanobacteria. And it is also found in a plant called cycad. And cycad plant contains a lot of these BMAA, and particularly its fruit. Now, it has been discovered by Cox and his colleagues back in 1997 that there's a huge amount of BMAA in the brain of Alzheimer's patients. And then that led him to go to a island, which is a Guam island, where there is a special tribal people they call Chamorro people, and they are suffering very high number from a particular dementia, which they called Parkinson and Alzheimer's disease combined. And he investigated these people extensively, and they found that there is a huge amount of BMAA in their brain tissues. And following that observation, he made many trips to that island, and ultimately he concluded that these particular neurotoxin in the environment might be the causative agents for the high level of neurological diseases that have been found in that island. Now, another source is cyanobacteria. This is a green algae, cyanobacteria, that we see in local ponds, but these are found in the ocean floor. And these are very special type of cyanobacteria, and that produce BMAA. And there is a scientist, his name is Hans Perl from the University of North Carolina. And many years back, he noted that this particular cyanobacteria is the source of high level of BMAA. And this is a
what you can
video that
see, obviously, is that it's a very thick population of algae. And in fact, when they form the blooms and lakes, they get even thicker than this. It looks like a carpet that kind of undulates out there on top of the water. It's almost like an artificial golf course kind of surface. These organisms grow as like a foamy, sort of olive green, not unlike my shirt, color to it. And in some ways, it has kind of a beauty to it. Cyanobacteria are microorganisms that are pigmented. They have the pigment in them that all algae have in them, the green pigment called chlorophyll. But they're different from regular algae because they produce a wide variety of organic compounds, some of which may be toxic or make animals ill, ranging all the way from the tiny plankton that eat algae to fish to animals like pets and dogs and cats and cattle that might drink the water, and even people. They've become really a more serious problem probably in the last 50 years or so, or the post-World War II boom in population growth, increases in agricultural expansion, urbanization, and industrialization, all of which has put a lot more nutrients into freshwater systems, rivers, and even in coastal marine systems. And that has brought about some of these very large blooms. It's difficult to clean a lake up of these organisms. They're microscopic. So really the management strategy is to reduce the nutrients that are coming in to cause the problem in the first place.
So you have seen that this particular cyanobacteria, which is grown on the ocean floor, could be a huge source of BMAA. And in fact, there have been a huge amount of work that has been done on the coast of Florida. And they have mapped out the entire ocean floor where these particular cyanobacteria grow. And therefore, they instruct the divers not to go there, not to expose themselves too much to these particular type of cyanobacteria. Now, these type of cyanobacteria has also been found in other parts of the ocean. Now, so cyanobacteria is one source of BMAA, and the other source is cycad fruit. And I mentioned about these tribal people who are living in a remote part, and this is actually the area which you cannot see. There are many islands here. It is in the Indian Ocean. Now, I think it is in the Pacific Ocean. And there are many, many tiny, tiny islands. You can see these are the islands. And in these islands, these Chamorro people live, and there is almost 50% of the people they're suffering from some kind of neurological disease. And the cause is BMAA. And because these people, they eat this particular fruit, which is called cycad fruits, and these cycad fruits, they make flour from that, and they make bread, and that is their main food. And so they are consuming a lot of BMA through the fruit. And on top of that, they also eat the bat meat, and the bat eat also these foods. So there are two ways they are taking a lot of BMA into their body, and the BMA level has been found to be extremely high in the brain tissues of these Chamorro patients. And their disease is initially called PDC, which is like Alzheimer's, but not 100% exactly identical with Alzheimer's disease. And what is the rationale then? What are the causes of Alzheimer's disease? So there are many causes. You can see that there are neurotoxins in this particular case, BMAA, and there might be many more neurotoxins in the environment. And this is our theory. So this is your tau protein. And this is your microtubule that is present in the neuronal axon. And this is how it binds. So there are 4 domains in tau protein, and these are involved in the binding with microtubules, leading to the healthy functional neuronal activity. So when caspase-3 cleaves here, it becomes a little bit shorter. And also when it becomes nitrated and phosphorylated, so these are the Y are the tyrosines which are nitrated, and these are the serine which are heavily phosphorylated. So the phosphorylation and nitration of the truncated tau protein is the cause, because it now loses its binding property with the microtubule. So there's no more binding with microtubules. And as soon as you lose the microtubule binding, then the neurons, they become unhealthy and they die. And it has also been found that the BMAA, this has been done in our laboratory, they have found that the BMAA level can be detected in the tissues using what is called reverse phase HPLC technique. So you can even detect 0.005 microgram of BMAA in a tissue, and it can be also detected by mass spectrum. But HPLC is a highly sensitive procedure or tool by which you can detect the BMAA level in the brain. And this is also done in our laboratory. We found that by adding BMAA to a peptide, it can lead to the formation of high polymeric peptides. So you can form dimer, trimer, tetramer, and so on. So that means the BMAA can trigger peptide polymerization or aggregation. Now, there are many other small molecules have been discovered in the later on. One is called DAB, another is called BOAA, and they have been linked to other neurological diseases. So you can see that these neurotoxins, which are in the environment, people should be aware of that and should avoid being exposed to these chemicals. And we have done in our laboratory that using some cell culture experiments that, for example, here, this is a transgenic mice. That means a mice which is a mimic of Alzheimer's disease, and it has been created in California, and by knocking out 3 genes. So if you knock out 3 particular genes, then that mice becomes like an Alzheimer's mice. That means this mice displays all the phenotypic features of Alzheimer's disease. And you can see that the corresponding wild type, that is normal mice here. Normal mice, there is very little tau, but in the case of Alzheimer's mice, you have a huge amount of tau protein, which has been aggregated. And then there's a huge amount of phosphorylated tau. And also there's a huge amount of nitrated tau compared to the wild type. So this explains that there is a there is a promotion of high level of phosphorylation and nitration. So now the question is intervention. So we have learned a lot about this disease, and we see that the protein aggregation is one of the primary causes of this disease. And there are some neurotoxins which can play in this event. So what are the ways we can intervene in the disease? So one of the ways to blocking the effect of BMAA from the environment, blocking caspase-3 activity, blocking kinase activity, because kinase are the enzymes that are responsible for phosphorylation, high amount of phosphorylation in tau, and also inhibition of tau nitration. So all these could be strategies in order to stabilize the disease so that it don't deteriorate further. So here are some of the drugs that have been available in the market and that are being approved to be used for Alzheimer's patients. And basically they are all copper chelating agents. For example, cleoquinol, this is actually used for ER infection. And later on, it has been found that it can be used. It is just an iodo-chlorohydroxy quinoline compound. And the structure is, you can find out in the literature, it is a very small compound, and it binds with copper. And later on, galantamine, which is a plant-derived natural product, and it has been found to be a copper chelator, meaning that it binds with copper. So that means it can remove copper from the brain or from the body. And that's why galantamine is prescribed for Alzheimer's patients. Azurin, which is a protein, and it is a copper binding peptide, and it inhibits aggregation. Then just simple arginine, which is an amino acid, it is also shown to be a blocker of protein aggregation. Then the highly used drug is actually Namenda, and this has been discovered as the first drug to treat Alzheimer's patients. But this drug is not blocking some of the issues that I mentioned in this talk. That means it is not something to do with protein aggregation. Actually, it is blocking the excess of glutamate. Glutamic is another player in this disease, and this is the earliest drug. And then you have Adicept, which is blocking another enzyme, which is called acetylcholinesterase activity, which I also did not discuss in these two. But these two areas, that means glutamic acid receptor and cholinesterase, these are the original areas where people were putting a lot of attention. But now the attention has been shifted to protein aggregation, because protein aggregation appears to be the major factor in this disease. And very recently in Canada, in Vancouver area, a new drug, which is still not approved yet, but it is in the final stage of approval. And this is developed by the Anavex Life Sciences, and it slows down the progression of AD. And it targets protein misfolding. Protein aggregation is also a part of misfolding. So therefore, this particular compound blocks misfolding of protein. So with that, I will stop here, and I will also like to show you that there is a video created by the University of Calgary. I want you to go through that slide later on, where you will see the role of mercury in protein aggregation.
How mercury causes brain neuron degeneration.
So I will not spend time going through this video.
Mercury has long been known to be a potent neurotoxin.
Just a little part of it.
Whether it is inhaled or consumed in the diet as a food contaminant. Over the past 15 years, medical research laboratories have established that dental amalgam tooth fillings are a major contributor to mercury body burden. In 1997, a team of research scientists demonstrated that mercury vapor inhalation by animals produced a molecular lesion in brain protein metabolism, which was similar to a lesion seen in 80% of Alzheimer diseased brains. Recently completed experiments by scientists at the University of Calgary's Faculty of Medicine now reveal, with direct visual evidence from brain neuron tissue cultures, how mercury ions actually alter the cell membrane structure of developing neurons. To better understand mercury's effect on the brain, let us first illustrate what brain neurons look like and how they grow. In this animation, we see 3 brain neurons growing in a tissue culture, each with a central cell body and numerous neurite processes.
So I will stop here, and at the very end, I wanted to look at this particular video, which has been created by the Alzheimer's Society of Canada. And the link is there, and you will see what are the different features of Alzheimer's disease and what people should be careful about in order to avoid catching this disease. So I'll stop here, and I will appreciate if there is any comments and questions from the audience. Thank you so much, and I really appreciate having this opportunity to share some of our own work, as well as a lot of research that has been done globally, and what we know about this particular disease, which is spreading so much worldwide. Thank you.
Thank you, sir, for taking out time from your busy schedule and sharing your insights on such an interesting topic. The lecture was really informative, and I'm sure we all learned something new today. Now, I would like to put forth a few questions asked by the audience. The first question is, where does the excess of Cu2+ ions arise from in the brain tissues? Is it due to malfunctioning of copper storage proteins like ceruloplasmin?
Well, this is one theory that high level of copper ion, and copper is an oxidizing metal ion. So it can cause some oxidation of the proteins. But the main role of copper ion, I think, is the promotion of self-aggregation that we do not understand completely, but all we know that copper forms complex with A-beta-42 peptide, and therefore copper must have some role, and removing copper using copper chelating agents can be a treatment, and it is a treatment. So therefore, copper being a oxidizing transition metal ions, as well as forming complex with A-beta-42 via the histidine and tyrosine amino acids. So those are the basic connections between high level of copper. And also copper has been found in high level of brain tissues, mortal brain tissues of AD patients. So therefore, copper must have some role in this event. Thank you.
Thank you, sir. Moving on to the next question. Is Alzheimer's dementia caused due to some mutation in chaperones, which causes improper protein folding and aggregation?
Yes, the answer is yes. As you can see that there are almost 50 different mutations have been found in APP, in human brain. And some of these mutations are highly detrimental because they can lead to the formation of high level of A-beta-42 peptide. And as I mentioned, Swedish mutation. And after Swedish mutation, you have Finnish mutation from Finland. And they can cause high level of beta gamma secretase activity. And because of that, you produce a large amount of A-beta-42 peptide compared to normal AD patients. So therefore, mutation must have a big role. So this is a genetic part of the disease. Thank you.
Thank you, sir. The next question is, why the Swedish mutation causes the beta secretase cleavage 9 times faster?
The reason is that beta secretase enzyme, this is a hypothetical name. Now it is called base enzyme. Now, base enzyme has been identified, it has been characterized, and its enzymatic characteristic properties have been well studied. And because of these KM mutation, K is lysine and M is methionine, the enzyme recognizes the site 9 times easier. So 9 times better. As a result, there is a 9 times faster cleavage. So it is because of this motif of this enzyme. Thank you.
Thank you, sir. The last question is, Can we synthesize artificial proteins that reactivate the neurons in an Alzheimer patient?
Yes, that's a good question, because there are some peptides that have been discovered, and actually there is one peptide which is commercially accepted and approved to be used, and this is called a peptide which is coming from a beta-42 peptide from the middle part. From the middle part, there is a hydrophobic core. So that hydrophobic core peptide, it is a short 5 amino acid peptide, and that peptide is currently used as a drug because it can block a beta-42 interaction. So it is a competing peptide. So it is a peptide that blocks interaction, self-interaction between two A beta-42 peptides. So to answer, yes, peptides have been very useful in this field. And as I mentioned, there's one peptide already been in the market to treat Alzheimer's patients. Thank you.
Thank you, sir, for patiently answering all the questions of our audience. Now, I would like to call upon Dr. Reena Ghosh to deliver the official vote of thanks.
I'm sorry, but the vote of thanks is not mine. I would like to address the MC. There has been a mistake.
Yes, ma'am. Yes, ma'am. I'm sorry. Dr. Rahul Sharma, Associate Professor, Department of Chemistry, St. Xavier's College, Autonomous, Kolkata, will be delivering the official vote of thanks.
Okay. Honorable Professor Basak, respected colleagues, and dear students. It is my privilege again to have been asked to propose the present vote of thanks on this occasion. I, on behalf of the Department of Chemistry, St. Xavier's College, Kolkata, and the entire organizing committee of RACB'22, all the attendees, and on my own behalf, extend a very hearty vote of thanks to our speaker, Professor Ajoy Basak, for sharing his findings and informing us about the work done across the globe. Thank you, sir. I express my gratitude to Father Principal and Vice Principal. Without their patronage, this event would not have been possible. I would like to acknowledge our gratitude to the day's chair, Dr. Reena Ghosh. Thank you, ma'am. We thank our audience for their cooperation and enthusiasm that was crucial for the success of the event. Finally, I would like to take this opportunity to place on record our heartfelt thanks to the members of the organizing committee, the students of our Department of Chemistry, for their utmost dedication in organizing RACB'22 Chapter 2. Without their hard work, this whole program would not have been possible. Thank you. Thank you all. Good evening.
Thank you very much.
With this, we come to the end of today's webinar. Hope all of you have learned a lot. I would request all the participants to fill up the feedback form rolled out in the Zoom and YouTube live chat within the stipulated time. With this, the journey of RACB'22, a series of international webinars, comes to an end. The organizing committee is highly privileged to conduct these webinars and share the platform with such eminent scientists. We thank our audience for their constant support and encouragement. The Department of Chemistry, St. Xavier's College, Autonomous, Kolkata will be back again with such engaging sessions. Till then, subscribe to our YouTube channel for getting further updates. Good night and take care.
Thank you.